The overall aim of this protocol is to efficiently target transgenes to loci of interest in human embryonic stem cells using custom-designed zinc-finger nucleases. To do this, human embryonic stem cells, or HESCs, are expanded to generate enough material for electroporation, then digested to yield single cells. Next, mRNA encoding two custom-designed zinc-finger nucleases is co-electroporated into the HESCs with the linearized EGFP donor sequence, and cells are then plated onto fresh mouse embryonic fibroblasts, or MEFs. The electroporated zinc-finger nucleases introduce a double-strand break into the first exon of the PIT-X3 locus in the cells. Then, the EGFP donor sequence, which is flanked by targeting arms with homology to the DNA double-strand break site sequence, is incorporated during cell division. After 14 to 21 days, 1/3 of an individual HESC colony is passaged onto each well of a matrigel-coated 96-well plate, and the other 2/3 is transferred to a corresponding well of a 48-well plate containing fresh MEFs. When the cells on the 96-well plates reach confluency, they are screened for incorporation of the donor sequence by PCR. Southern blotting is then performed to confirm correct integration. Once positive clones are confirmed, they are expanded and then frozen for further use.
The main advantage of this technique over existing methodology, such as conventional and homologous recombination, is that zinc fingers create predetermined DNA double-strand breaks.